Now we will see the next 3 marks question, that is question number 35, which is again divided into sub question A and B. First we'll see A. A short pea plant, that is small t, it is pure one, homozygous, is crossed with tall pea plant, that is capital T, this is also homozygous. Express the result obtained in the F2 generation with the help of checkerboard. So very simple question. So see here what you have to do. First you have to cross these plants, that is pure short pea plant and pure tall pea plant, and first find out what is F1 generation by cross pollination, and then by using the progeny of F1 generation do the self pollination and find out F2 generation with the help of checkerboard. So see here small t both are recessive. So here this is called as pure. Capital T, it is called as mixed, or you can call heterozygous. Remember, heterozygous. This is homozygous. It is not mixed, it is pure, homozygous. But here this is also pure. Sorry, I wrote it wrong. Don't get confused. Sorry for the mistake. This is also pure and this is also can be called homozygous because both are capital T, capital B. But if one is capital T and one is small t, this is also showing the tall characteristic, but it is not pure, it is mixed. You can see here one is capital T, one is recessive, one is dominant, so it is mixed. This is called as heterozygous. I hope you understood, don't get confused. So here small t pure also called homozygous, capital T pure also called homozygous. This is dwarf and this is tall. Now you see this one, capital T, small t, this is also showing a tall characteristic, but it is not pure. This and this has a difference. Here it is mixed, or we can say heterozygous. So here we will start the answer. Now we have two plants, so I'll write it here. Short pea plant, and then here I will write tall pea plant. These both are pure, both are homozygous. So I write it T and T, capital T and capital T. Done. Now these both the plants are different. Remember, this is different plant. So now if you do pollination in this, it is called as cross pollination. So first write down the genes, small t1. Now see here, capital T1. Because both are same, we can write only once. Once we cross these two, such type of pollination is called as cross pollination. I hope you understood why it is called as cross pollination, because both the plants are different. This plant is different, this plant is different. When we cross these two, we are going to get a progeny with capital T and small t. Now see, this will show the character tall. All the progeny in this generation, this is called as first generation, that is F1 generation. So in F1 generation, whatever progeny we are going to get, all will show tall character, but this is not pure. One is capital T, one is small t. Therefore, this is heterozygous. This is heterozygous, or we can say mixed. It is not pure. But these both were pure. This was also pure and this was also pure. Now by using the progeny of this F1 generation, we are going to do F2 generation. So let me divide the page into two. Now here we will start with F2 generation. In F2 generation, we are going to use the progeny from the F1 generation. So we will write it here. Tall pea plant can be shown by capital T and small t. Same plant we are going to use it. So tall pea plant, once again I'll write it. Capital T, small t. Now see, these are not pure, these are mixed. These are mixed, or I can say heterozygous. I hope now you are understanding what is heterozygous and homozygous. So now write down the genes. So see, because it has two different, one is dominant, one is recessive, we are going to get two, capital T, small t. Here also we are going to get one capital T and one small t. Now if you see, these both the plants are same. We are going to do the pollination in the same plant. Capital T, small t, capital T, small t. So this type of pollination is called as self-pollination. Because both the plants are same, or you can say in the same plant we are going to do the pollination. So when we cross-pollinate these, self-pollinate these two plants or these two genes, we will get the progeny of F2 generation. In the question, they have asked to write this F2 generation with the help of checkerboard. So I'll draw the checkerboard here. So I'll write it here, checkerboard. Now, so this is called a checkerboard. Write on the gene capital T, small t, and here I have to take one more row for this. Let me take one more row here. Now write down capital T, capital T, and this is small t. So first one is for this one and second one is for this one. Now we will cross it. So see here, capital T, if you cross, what you're going to get? Capital T, capital. Now this capital T and this small t, if you cross, then you will get capital T, small t. Now this and this, if you cross, you are going to get capital T and small t. And last is this and this, if you cross, you will get small t and small t. So this we got the result of F2 generation with the help of checkerboard. Now if you write the physical character, capital T means we are going to get pure tall pea plants. Capital T small t means it is also tall, but it is not pure, it is mixed. This is also tall but mixed, and this is dwarf, that is pure. So this is the phenotypic character or character in this F2 generation. Now we will write phenotype and genotype ratio. So I'll write phenotype and genotype ratio this side. So I'll write phenotypic ratio. Phenotypic ratio means what? By seeing the physical character, we have to write the ratio. So tall. First I'll write tall. Then I'll write dwarf. So how many tall plant we got? We got total 1, 2, and 3. So I'll write 3. Is 2. Dwarf is how many? Only 1. So I'll write 1. So this is the ratio that is called as phenotypic ratio. Now I'll write it here genotypic ratio. Genotypic ratio. In genotypic ratio, we have to see the genes. So we have 3 tall, but in these 3 tall, 1 is pure and 2 are mixed. So therefore, T and T I'll write once is 2. Capital T, small t I'll write is 2, small t and small t I'll write. Now see, capital T how many? Only 1. Capital T, small t how many? Are 2. And small t how many? 1. So this is the called as genotypic ratio for this F2 generation. So I hope you understood the answer for this question, that is 35, sub question A. After this, we'll move with the next question. Now we will see the sub question B. How is baby girl sex will be determined? So see here the question, sometimes how the sex in the new individual will be determined in the human being. Like this will be question asked, and sometimes it will be like this. But the answer for both the question, it is same. So see here answer. The sex of a baby is determined by chromosomes of the parents. So here always remember the sex of the baby is determined with the help of chromosomes which are coming from the parents. The mother has 2 X chromosomes, that is XX, and the father has 1 X and 1 Y chromosome, that is XY. So as we all know, total chromosomes we have, that is 23 pair. We have 23 pair of total chromosomes, and out of this 23, we have 22 pairs, that is called as normal chromosomes or autosomes, and 1 pair we have that is called as sex chromosomes. So this you have to remember. So what do you mean by autosomes? These are the chromosomes which are going to help the individual to decide the normal characters, like it may be a skin color, or it may be a length of the hair, or it may be height, etc. So these all are normal characters which will be passed from the one generation to next generation, and that all will be controlled by these autosomes. But the new individual will be a boy or a girl. This is depend on this sex chromosome, and it will be a 1 pair. And remember, in females, in mother, that sex chromosomes will be XX, and in father, that sex chromosome will be X and Y. If the father gives X chromosome to the baby, the child will be a girl, because in mother both are XX. So always the egg is going to have, all the eggs are going to have X chromosome. But in father, as the X chromosome consists of X and Y, some sperms will be having X, some sperms will be having Y. So therefore, if the father gives X chromosome, then the X and the X from mother, it will become a girl child. If the father gives Y chromosome, then Y and the X from the mother, it will become XY, and the child will be a boy. So let me explain you in detail. So I'll write it here, father, and here I'll write mother. So what is the X chromosome in father? It is X and Y. In mother, it is X and X. So all the eggs in the mother will be having X chromosome as a sex chromosome. In father, some sperms will be having X chromosome, some sperms will be having Y chromosome. Now, whenever the fertilization takes place, sperms and egg fuses. If the sperm having X chromosome fuses with the egg having X chromosome, then the child will have XX as a sex chromosome. And if it is XX, then remember the child will be always girl child. If the sperm having Y chromosome fuses with the egg having X chromosome, then the sex chromosome will present in the young one, in the child will be XY. Then the sex of the child will be a boy. So this is the main thing which happens during the fertilization. So what this shows, therefore, a baby girl is born when the father contributes an X chromosome. So remember, the child will be a boy or a girl. It is depend on the sperm coming from the father. If the sperm is having X, then the child will be girl. If the sperm having Y will come, then the child will be a boy. So this is the answer for this sub question B. I hope these answers will help you to prepare for your final exam and take good marks in your final exam. If you like my videos, please share my videos with your friends. And if you have any doubts, comment those doubts in the videos. And if you are not yet subscribed my channel, please consider subscribing my channel. Thank you so much.